pine tree looking arrangement. It looks almost like the growth rings of a pine tree. There's a central canal right in the middle, and then there are these concentric rings. They're called the lamellar rings. And the rings have these bone cells in them. And we're going to want to say a little bit more about that here in 1 second. Okay, so this gives the strength to the bone. Compact bone is really strong. It's the kind of strength that you would find if you took a single pine tree and put it in a hurricane, or you let a half dozen pine trees grow up all together. Try to push that over. It's that kind of strength that the compact bone gives. The nature of the compact bone here, its strength. Okay. Now, notice that in the epiphysis of the bone, there's a thin lining of compact bone on the edge, but the bulk of the mass of the epiphysis of the bone is actually spongy bone. It's sometimes called trabecular bone. And these trabecula are really it's an interwoven network of bony spicules that create caverns. It's lighter than compact bone. So, like the mastoid process of your skull, for example, is mostly spongy bone. And the skull itself, although it's very strong, it has spongy bone in the middle of it. It's compact on the inside and outside. It's like a sandwich of spongy bone. If it were not so, your skull would be so heavy that the musculature in your neck to support the weight of your head would make all of us look quite funny. So our heads are heavy enough as it is, so the spongy bone helps take some of the weight off. There's some more importance to be said about the spongy bone here. The spongy bone, as you've already learned in this class, is also the house of bone marrow. Now, as I work my way down into the shaft of the diaphysis, who's that? Oh. The shaft of the diaphysis of the bone, you can see that there's a thin lining of spongy bone within the shaft even of the long bone. But the distribution has changed. You have a lot more spongy bone in the epiphysis than you have compact bone, and you have a lot more compact bone than you have spongy bone in the diaphysis. Now that makes good sense. On the ends of the bones you have strength on the edges, but in the middle you have weight reduction, and in that trabecula, in the ends, in the epiphyses, you have red bone marrow. This is an important location for blood cell development. So this area here, this epiphysis of this bone, contains red bone marrow. And as we've already pointed out, if you took a section of that and looked at it in a microscope, you should already have an idea what that looks like. That's reticular, right? Scaffolding, blood cell scaffolding with the collagen fibers, reticular connective tissue in the ends of the bone. Compact around, spongy, a little spongy here. Now the marrow here in the diaphysis of the bone is yellow marrow, and we saw that too. That's adipose tissue. So there's red bone marrow, reticular tissue in the epiphysis. There's yellow bone marrow, adipose tissue in the diaphysis. The bulk of the shaft is compact. The bulk of the epiphysis, the bulk of the end of the bone, is spongy bone, and we find red bone marrow there. Now, you can also see a couple of important membranes. There is, in fact, a periosteum that covers the bone that we don't have. Periosteum is a dense fibrous connective tissue, and the periosteum of bones normally is the living connective tissue sheet that fuses with the tendons and ligaments that attach to bones. So you might have wondered to yourself before, now that you started anatomy, whenever a muscle is attached to a bone, how exactly does that happen? How does the bone hold on to the other bone by way of a ligament? I understand there's a ligament across there. I understand there's a tendon that goes to the bone, but how does it fit on the bone? Those tissues fuse in with the periosteum. And the periosteum, that living membrane around the bone, is actually attached to the bony matrix by way of tethers that you will never be able to dissect the way this picture shows. The reason I know that is because I have tried it in cadavers. It cannot be done. This periosteum here is linked to the bony matrix by way of perforating or Sharpie's fibers that hold the periosteum to the bone. It's like an uncountable number of staples in which each staple is a linkage from the membrane into the bone, and each one is gram for gram stronger than a piece of steel. So to say that the attachment is strong, you see, is an understatement. The periosteum and its connection by way of Sharpie's fibers anchoring to the bones and fusing with the tendons and ligaments that anchor to bones create an incredibly strong connection. Okay, so I introduced periosteum and Sharpie's fibers in the marrow and the compact and spongy bone. All right, the next tissue, so we've moved from the connective tissue propers to the cartilage to the bone, from the fibroblast tissue to the chondroblast tissue to the osteoblast tissue, and finally to the hemocytoblast tissue. This is the one that we don't think of as tissue unless you see it in its clotted form. Now, the major protein that we talk about as being fiber of connective tissue is collagen. The major protein that we talk about as fiber in blood is the protein fibrin, Fibrin. Fibrin is the insoluble protein that creates clots, blood clots. And so that's really the final piece of describing blood as a connective tissue. Now, for this class, I'm going to ask you to know just a little bit more about blood. First, I'd like you to know that blood is made up of two components, two primary components. It has a cellular component, and that cellular component is made of cells. Those cells all arise from stem cells called hemocytoblast stem cells. So all this was on that original picture of the connective tissues. Hemocytoblasts forming the different blood cells. So the cellular nature, the cell component of blood, is that all these cells are made from the same stem cells. So whether it's a red blood cell, a white blood cell, or a platelet, let me say the science words, whether it's an erythrocyte, red blood cell, or a leukocyte, a white blood cell, or a thrombocyte, a platelet, they all come from the same hemocytoblast stem cells. And the cellular nature of blood comprises, on average, 45% of any given blood sample. And of that 45% of the blood sample, more than 90% of it is red blood cells. So in physiology next semester, I'm going to give you a capillary tube that has heparin in it, so the blood won't clot. And I'm going to ask you to prick your finger and use capillary action to put blood within the capillary tube. And then we're going to take those tubes and stop them up with clay on the end and put them in a centrifuge. And we're going to spin them really fast for about 5 minutes. All of the cellular nature of the blood is going to pack on one side, leaving just the clear plasma on the other side. And we'll measure that percentage, and it will make up about 45%. We call that the hematocrit. Hematocrit, the hematocrit. 45% of the blood sample, the hematocrit. It's the cellular component of blood. And more than 90% of it is red blood cells. Now, from your past educational experience, what is the major function of a red blood cell in the human body? What do you need them for? What do red blood cells do? Great, thank you. I was going to say, my goodness, I've got some work to do. All right, so they transport oxygen in your body, right? So if you take your hematocrit, you spin it, and the hematocrit number is 30% instead of 45. Can you tell me what the life experience of this individual is going to be? You know without asking them. What are they going to feel like? The cellular component is dramatically lower. What do they feel like? So I've had this happen in the lab before. I had a young lady who was pregnant who had had my class. She said, before I go to the doctor, I want to come by here. So she went in the lab and did her hematocrit. It was like 32. She said, well, that explains why I feel so faint, so tired, so non-energetic, so without life. Yeah? The cellular component is dramatically down. Okay, so that's the cellular component. The other part of the blood is the plasma, and it's mostly water. So remember our characteristics of connective tissues. Cells, let's see, can we name them? Connective tissues are made of cells. The connective tissue propers, the primary cell type is fibroblast. Cartilage, the primary cell type is chondroblast. Bones, the primary cell type is osteoblast. Yeah, so this was that first slide that we looked at to distinguish all the tissues. Fibroblast, chondroblast, osteoblast, and here, hemocytoblast. So we say the tissues have a cellular nature. And we talked about the cells as being the cell-producing tissues. So you've heard that with each of these tissue types. And then we said, outside of the cells, there's this non-living extracellular matrix. Now, is the non-living matrix just simply a goo? Is it simply something that we don't understand? Is it something that we don't know what is there? Why does some connective tissue have a hard feel to it and some of it a softer feel? What's in there? These are proteoglycans, yes, with different content of hyaluronic acid and chondroitin sulfate. Y'all remember that story? That make up the consistency of the tissue in the matrix. Now, does the plasma, in quotes, the matrix of this tissue, does it have fibers in it? Let's start with the other. Does a dense regular connective tissue have fibers in it? Does a loose areolar connective tissue have fibers in it? Does hyaline cartilage have fibers in it? Yes. What's the primary fiber? Collagen, right. The protein collagen, the main structural protein of the human body. So when we get here, we look and we say, I don't see any collagen there. Does blood have collagen in it? It better not, because this has got to be liquid. It's got to flow through the human body. So is there no fiber type? Why do you keep calling it a tissue? So you know the answer to this. There's a protein here that is solubilized. It's soluble in the plasma. You don't see it. But when you have a clot that forms, that protein will come out of solution, and you will see that blood is in fact a tissue. And fibrin comes out of solution. All right? Now here's some white blood cell types. Red blood cells are really easy to find. They're everywhere. These biconcave little discs here. The white blood cells are really the histological work. If you want to diagnose leukemia, then you'll want a sample. Let's just work through this, see if you guys are following me. I want to diagnose leukemia. I'm a pathologist. The leukemia is going to be diagnosed by finding abnormal cells during development. If it is a leukemia, what kind of cell am I going to be targeting? Red blood cells, white blood cells, or platelets?
White blood cells.
White. White blood cells, the leukocytes. Yes? What kind of tissue would I want a sample of in order to see the developing white blood cells? Where do blood cells develop? In the marrow. What kind of marrow? Red bone marrow. Where is the red bone marrow? In the ends of the long bones. You'll start to put these things together, right? This is anatomy that should be interesting to life here. Now, there is also red bone marrow in spongy bone in other locations. Some of you know this already. You've had friends, family, who've had bone marrow samples taken from them. Does anyone know? Where is the most common site for getting a sample of bone marrow? Anybody know? Had anybody that's, none of you have had any friends or family who's had marrow taken? They want to diagnose the leukemia. They want to look at the white cell development. The most common place is in the hip. Now, the skull has a lot of red marrow in its spongy bone, but you don't want to be sticking needles into the skull bone. The sternum is sometimes used to get samples of red marrow, but that could be dangerous. Could be, right? Because there's some rather important structures around there. The hips are a very popular place to get marrow samples. And so they use large gauge needles to insert through the articular cartilage and the compact bone into the spongy, and then they extract marrow samples. It's a very painful procedure. And then the pathologist looks for these guys. Looks for the abnormal development of the white cells. This is what normal white blood cells look like in a blood sample. So you can see the types: neutrophil, eosinophil, basophil, lymphocyte, and monocyte. These three up here, named for the way they stain. Neutral, acid, base. This is the way they stain. It doesn't have anything to do with their function. It's the way they behave in a hematoxylin stain. The neutrophils, eosinophils, and basophils are what we call the granular leukocytes. And you see why. Well, certainly you can see them here. And the eosinophils and basophils, the granules. And this cartoon does not show you granulated neutrophils, but they are. So these three are typically filled with granules in their cytoplasms. Neutrophils, eosinophils, and basophils. These two, the lymphocytes and the monocytes, are agranular leukocytes, and so their cytoplasms do not have granules, or at least small, at least not dense granules, in their cytoplasms for visualizing under a microscope. Now, in physiology, we're going to walk through each of these, and I'm going to tell you what they do. But for now, I wanted you to see them and get some categories in your head. Granular and agranular leukocytes, the 5 major white blood cell types that you find. All right, the next tissue type, as we're almost done with tissues, is muscle. And there are 3 varieties, and there are some anatomical peculiarities about each of them that will help you identify them under a microscope. The first one here shown is skeletal muscle. And skeletal muscles are striated. Now, in anatomy, if I want to explain in anatomy why something looks the way it looks, or feels the way it feels, has the structural capacity that it has, what organic macromolecule am I going to use to describe that? Yeah, we've said this so many times, right? So when I first look at a skeletal muscle section, I notice that the cells are striated. They look striped. The reason for those striations is the arrangement of 2 proteins, primarily. There are actually 4 here. We'll do the tropomyosin troponin story later, but 2 primary proteins that explain why these things look striated. They are actin, A-C-T-I-N, and myosin, M-Y-O-S-I-N. Actin and myosin create the striated appearance of skeletal muscle. Now, this says that the skeletal muscle has multinucleate cells. And there are some, I know some histologists, Dr. Du Bois may do this, I don't know, who ask their students to isolate individual cells by trying to find their borders, which is difficult, especially in a histosection, because you might have a skeletal muscle cell that extends several centimeters, so that can be complicated. And then having the students locate multiple nuclei within each cell. I don't like that technique, and I don't think it's the best one for identifying skeletal muscle anatomy. If you compare the arrangement of the proteins in a skeletal muscle cell to the arrangement of the proteins in a cardiac muscle cell, you will find that they are the same. Cardiac muscle cells and skeletal muscle cells at high magnification cannot be distinguished from each other because the arrangement of the actin and myosin protein is identical. Now you can see why a histologist would go after the nucleus story here, because the cardiac muscle cell is said to have a single nucleus per cell. But I'm going to give you something easier anatomically to be able to distinguish these. A student should be able to look at this image and this image and immediately notice something dramatically different. Between this image and this image, something jumped out at you. These are not the same, and you could tell immediately. What was that? What's different when you look at this and you look at that? What jumps out at you? Can you tell they're different?
Mm-hmm.
Okay. How did you tell?
The little circle things and the white cloudy looking stuff.
Okay, so circle things. You're pointing at the nucleus.
Oh, yeah.
Okay, and what else did you say? The coloration.
Yeah.
So you know this one looks darker than this one.
Mm-hmm.
Okay. Well, it's a little unfortunate that the colors are that way because.
No, I said the white stuff.
Okay.
The
Yes.
thick looking stuff.
So you noticed here, and here, and here,
Mm-hmm.
and you didn't see that. Just a little thin sliver here, a thin sliver there. So you noticed that there seems to be larger gaps between them. All right, good. Anything else? How do these cells arrange with one another compared to the way these cells arrange with one another? Say again? Yes, we're gonna get there. Let's do big picture first. So this cell is running this way, yeah? That one is angled this way. That one is angled that way. They are, in essence, lined up, but many of the cells appear to be obliquely arranged with each other, whereas these here look more packed. So you're noticing the white between them. And I'm noticing, as adding to your observation, that they don't all run parallel to one another, which creates these gaps. So the easiest thing to look at in these is the arrangement of the cells. When you first look at them, it always jumps out at you, the arrangement. The cardiac muscle cells are not packed together like the skeletal muscle cells are. They run in this kind of oblique arrangement to each other, creating gaps between them. And those gaps are dense connective tissue and actually form, because of the collagenous nature of those structures in there, they form a skeleton for the heart. You know, when these guys contract, on this side and on that side, they're attached to a bone. So they have something to pull on. What does the heart pull on? It has to pull on the connective tissue that's laced between the cells. It has a skeleton. So the arrangement of the cells jumps out at you. And then what has already been pointed out here is the big one. And I introduced this to you and told you this picture was coming earlier in this class, cardiac muscle cells are actually connected by a special kind of junction called an intercalated disc. Now, from what you learned from me about cell junctions, you know, proteins that hold things together. Sometimes, like in the stratum spinosum of skin, desmosomes. Sometimes, like in the sheets of smooth muscle in the GI tract and the blood vessel walls of the brain, they're zipped together with tight junctions. And in other places, proteins link cells together, creating holes so that one cell cytoplasm is continuous with its neighbor. What kind of cell junction would we call that? A gap junction. And that's what these are. These intercalated discs are a telltale anatomical sign. You are looking at cardiac muscle here. And what this tells you is cardiac muscle cells are all connected together. All the atrial cells are connected together, and all the ventricular cells are connected together, which means that these muscle masses, although they are made up of thousands of cells, behave as though they are one cell. We call that a myocardial syncytium. The syncytium, the single, they are working in sync. They are one cell. This is why the heart is so efficient at pumping blood. You don't have motor units there that choose one cell or one group of cells over another one. Instead, you stimulate one, the whole mass gets stimulated, and it contracts as a single unit because of these anatomical junctions, the intercalated discs. All right, the last of the muscle before we do nervous stuff on Friday is the smooth muscle cells. These smooth muscles cells exist in, I don't have time. I got 2 minutes. I'm going to introduce it to you. I don't have time to tell you what I want to tell you about smooth muscle cells because I got some review to do with you here. Okay? So smooth muscle cells are found mostly in the walls of hollow organs. So when I start this, I didn't quite finish. When I start this on Friday, I'm going to talk about different kinds of smooth muscle and the junctions between them. It's going to explain the anatomy of a lot of places in the human body. So let me leave you with this. If there is a hollow place in me, let's say I go into the mouth and I come down the mouth into the esophagus and the stomach and I work my way all the way through the GI tract. That is a hollow tubular network. I expect to find epithelium lining it. I expect to find smooth muscle in the middle of it. And I expect to find connective tissue on the outside of it. If I go through here and I go into the airway and work my way through the trachea all the way through the alveolus, I expect to find epithelium lining it. I expect to find smooth muscle in the middle of it. And I expect to find connective tissue on the outside of it. If I manage to go into a vein and I get inside the cardiovascular system, I'm in the blood vessels, I am inside a lumen, I expect to find epithelium on the inside and smooth muscle in the middle and connective tissue on the outside. If I'm in a lymphatic vessel, if I'm in the urogenital system, if I'm in the kidney tubes, if I'm in the uterus, if I'm in the bladder, if I'm in the urethra, if I'm in a hollow place in the human body, I'm going to find this. So I'm going to introduce you to the different types. Your book doesn't do it here, but anatomy students need to know the details of this. And then we'll do smooth muscle, and then I'm going to show you some examples of nervous tissue. And then we're going to head toward the other introductory material that you're going to need to do the rest of anatomy. All right.